This is part 2 of 3 on chapter 11. In this chapter we're looking at physical and chemical agents for microbial control. So in the first part of this chapter we talked about the different terminology, and then we also talked about how some microorganisms have a higher resistance to being killed or slowed down compared to other microorganisms. So you have the high resistance, moderate, and low resistance. And we also covered microbial death, so how we determine if a microorganism is dead. So this section we're going to be looking at some of the physical agents. So if we look at our little chart over here, so I mentioned in the first part that you should be familiar with this chart. This part we're going to be looking at the pink part, so we're just going to be looking at physical agents in this part. Part 3 we're going to look at the chemicals and the mechanical part, the blue and green part of this chart. So we're going to start off looking at heat. So heat, we can measure this method a couple different ways. So we can measure thermal death measurements. So bacterial endospores, remember, are the most resistant to most of these control methods. So bacterial endospores, they usually require very, very high temperatures above boiling. So in order to determine if an organism is dead, we can look at the thermal death time, or the TDT. This is the shortest amount of time required to kill all the test microbes at a specific temperature. So thermal death time, you would pick a specific temperature, so let's say 100 degrees Celsius, and then you figure out how long it takes to kill all the microorganisms at 100 degrees Celsius. And then that's the thermal death time. So it's based on time. You can also have something called a thermal death point, or the TDP. This is the lowest temperature required to kill all microorganisms in your sample in 10 minutes. So this is different. Here you're controlling the time, so you only have 10 minutes. But you want to figure out what temperature you have to be set at for 10 minutes in order to kill those microorganisms. So one type of control method is heat. There's two different types of heat. There's moist heat. This is usually, you can have lower temperatures, shorter amount of time, and the heat, the way it works is it alters the protein structure. So that's the method of action. You can also have dry heat. Dry heat has to be a little bit moderate or higher in temperature. Dry heat usually works by dehydration. It can also alter the protein structure because you'll heat up your cell, your protein will be denatured. And then what we use in lab is the Bunsen burner. We use a flame. We're basically incinerating our bacteria, so you're just basically burning up the cells. So we use dry heat as our method in lab a lot of times. So moist heat methods. The most common moist heat method is using an autoclave. So this is where we have steam and it's under very high pressure. In this method, it's a type of sterilization, so you're killing all living things. In lab, I do have an autoclave that I use to autoclave all of the plates and stuff that we've used in lab. So an autoclave, it goes up to 15 pounds per square inch. So you have pressure, 121 degrees Celsius, so you have heat. And that pressure is produced by steam, so you have to add water in there. And then depending on what you're autoclaving, it can go from anywhere from 10 to 40 minutes. So this one you have a set temperature, and then you can change the time for the autoclave. You can also have non-pressurized steam, so tonalization. This is when you use intermediate sterilization for substances that cannot withstand autoclaving. So if you have certain objects that cannot go under the really high pressure, you can use this other type of moist sterilization. So your items are exposed to free-flowing steam for 30 to 60 minutes, and then they're incubated for 24 hours, and then you steam again for another 30 to 60 minutes, and you repeat this cycle for 3 days. This can be used for canned food. It can also be used for some of the laboratory media that cannot understand go under that high pressure. Another type of moist heat is boiling water. So boiling at 100 degrees Celsius for 30 minutes will destroy any non-spore forming pathogens. So this is when they talk about boiling water because it will kill all the bacteria and also a lot of the protozoans, so the animal-like organisms that can cause disease. Pasteurization, this is when heat is applied to kill potential agents of infection and spoilage without destroying the food flavor or value. So we use this a lot for juice or milk. So this is at a lower temperature, and you can do a batch method that's at 63 degrees Celsius for 30 minutes, or you can do a flash method, which is at a higher temperature for 15 seconds. So pasteurization, you're just getting rid of agents that could cause disease or spoilage in your food. Besides moist heat, you can also have dry heat. And again, this is at a higher temperature. So you can use incineration, so we use the Bunsen burner flame, or we have electric heating coils where you can stick your inoculating loop in there and it basically burns off all of the bacteria. There's also drying ovens if you want to sterilize other types of equipment. These are at 150 to 180 degrees Celsius, so they're usually at higher temperatures. So we talked a lot about heat. Cold is another method. This is a microbiostatic method. So this method, it just slows the growth of microorganisms. It doesn't actually kill the microorganisms. So this includes refrigeration. So we put our food in the fridge to keep it longer. And this is about 0 to 15 degrees Celsius. It's usually around 4 degrees Celsius. And then also freezing, which is less than 0 degrees Celsius, so less than freezing. These methods, again, just slow down the growth. So that's like, even if you put your food in the fridge, if you leave it in there for 2 or 3 weeks, it's going to grow something eventually. So this is just used to preserve food. I put our media in the fridge, usually if I remember to get it in there after lab. We can also put cultures in there to slow them down, which I try to pull out your plates from the incubator and put them in the refrigerator, because usually we don't want things to stay in the incubator for a week. So we use cold to slow down the growth. You can also do desiccation. This is where you gradually remove water from the cells and it leads to metabolic inhibition. So basically it just slows down the metabolic rate. This, for example, is like freeze drying things, which is used to preserve food. Or here, I order our bacteria, so like the E. coli, I order it, it's freeze dried. So the cells, they just have very, very slow metabolism. I have to rehydrate them to get them to grow. Okay, so we looked at different types of heat for physical agents, and also we looked a little bit at cold, how that kind of goes with heat. So the next part is looking at the radiation, the other side of the pink, physical agents. Alright, so we have 2 types of radiation. There's ionizing radiation. This has really deep penetration power, and it has a lot of energy in it, and it causes electrons to leave their orbitals in the atom. So these electrons, they start flying around and they start to destroy DNA or other parts of the cells. This type of radiation includes gamma rays, we have X-rays, cathode rays. We usually use this type of radiation to sterilize medical supplies, and we can also use it to sterilize food products. So here's an application of ionizing radiation. So these are raspberries. The raspberries on the left have been put under the radiation, so they're going to last longer, whereas the raspberries on the right were not put under radiation, so you can see fuzzy, that's actually mold growing on there. First I thought it was ice, but it's fuzzy mold because they were not put under the radiation. The other type of radiation is the non-ionizing radiation. This has little penetrating power, so it's not as strong. and you have to directly expose your microorganism to this radiation. An example of non-ionizing radiation is UV light, and this creates pyrimidine dimers. It causes your two thymines, if you have two thymines next to each other in your DNA, causes those two thymines to bond with each other and not to the adenine across from them. And then if you have two thymines bonded together in your DNA, it interferes with DNA replication. So basically the UV light damages your DNA, and then if the DNA is very, very damaged, it can cause those cells to become cancerous. So UV light is pretty dangerous. It's going to cause these dimers within the DNA. So not good for us, but you can use it to create these dimers in bacteria cells, and then those bacteria cells cannot replicate and they die off. So applications of non-ionizing radiation, we use it to sterilize air, we use it to sterilize water, or we can use it to sterilize different surfaces.